Alright, so this is the molecular basis of inheritance lecture on chapter 16. So here you can see the overview for the molecular basis of inheritance. We're going to go ahead and start with our nucleic acids. So nucleic acids, you've got DNA, you've got RNA. They're going to be made of nucleotides. You can see the nucleotide down here in the bottom right. Nucleotides are made up of phosphate groups, 5-carbon sugars, and nitrogenous bases. On the 5-carbon sugar, you can see that they are numbered. This is carbon 1, carbon 2. This is the 3' carbon, 4' carbon, and 5' carbon. That's going to be important when we start talking about how we're going to read the genetic material and how we are going to replicate it as well. Okay, so here we can take a look at the nucleotides, the nitrogenous bases specifically of the nucleotide. We've got adenine, thymine, guanine, and cytosine. A binds to T, G binds to C. And if you notice here, A and G, they are both double ring structures. These are going to be our purines, and T and C are single ring structures, and they are pyrimidines. So taking a look at the characteristics of DNA here, we can see that our DNA strands are going to run 5' to 3'. On the other side, it's running 5' to 3' as well. We'll talk about that in a moment. But each nucleotide is going to be consisting of this 5-carbon sugar, nitrogenous base, and then 3 phosphate groups, just like ATP. And then when they bind to the nucleotide strand to the DNA, DNA polymerase is going to use dehydration synthesis to cleave off the last 2 phosphates to create something called pyrophosphate. And that's going to be broken down into 2 inorganic phosphates. The energy that is released there is going to be used to help facilitate this process. So over here we can see that our strands, again, I mentioned, are running 5' to 3'. On the other side, it runs opposite 5' to 3'. So that is called anti-parallel. And when we're looking at each of these strands, one side is complementary to the other. So if I have a T on one side, I'll have an A on the other. G is always going to bind to C, C binds to G, and A binds to T. So when we look at DNA replication, it is going to be semi-conservative, and this was determined by Meselson and Stahl. They were looking at 3 different models. One was the conservative model, one was semi-conservative, and the other was dispersive. In the conservative model, it was thought that when the DNA replicated, you would have the original strand and a brand new strand. For a dispersive, you'd have a combination of new and old kind of randomly dispersed throughout. But it was discovered after experimentation that the semi-conservative model was correct, where each new strand has an old strand plus a new strand in the double helix of DNA. All right, so when we're looking at DNA, remember we're talking about chromosomes, and chromosomes are actually made up of DNA, which is this dark strand here, wrapped around these blue histone proteins. And when they're nice and tightly coiled, this is called heterochromatin, which is going to be very difficult to transcribe. In order to transcribe it, it's got to be a little bit more loosely packed so that we can actually get into the DNA in order for transcription to occur. Let's go ahead and take a look at DNA replication. It does occur in both prokaryotic and eukaryotic organisms. In the prokaryotic organisms, remember, they have a single circular chromosome. And so DNA replication is going to occur. It's going to start at one point and run in both directions simultaneously. It's going to be copying both strands. Okay, so it's going to have 1 replication bubble and 2 replication forks where the strand is actually being separated. In eukaryotic DNA, notice that we have a number of replication bubbles, each with their own pair of replication forks. Both sides are again going to be copied simultaneously, and then ultimately those replication bubbles will join together. The reason we have so many more replication bubbles in the linear eukaryotic chromosomes is because there's so much more genetic material, and it's a time saver. Okay, it makes it happen a bit more quickly. So let's go ahead and take a look at the replication fork in a little bit more detail. So here we've got several different proteins that we're going to be talking about. We've got helicase, you've got single-stranded binding proteins, RNA primer. All right, you're going to have DNA polymerase. But basically what's happening here, you've got a leading strand. The leading strand is going to be copied in a 5' to 3' direction. And this is going to happen continuously as this replication fork moves along the chromosome. On the opposite side, we have something called the lagging strand, and this is going to be copied in small segments called Okazaki fragments, named for the individual who determined how it functioned. And it's got to be copied again in 5' to 3' direction. So as this strand opens up, if this strand is copied and the leading strand is copied in a 5' to 3' direction, the other side is obviously, because it's anti-parallel, running 3' to 5'. So it has to be copied in small segments as that strand opens up. And this is going to involve a couple of other different proteins. In a moment, we'll talk about those. So here is your list of proteins you do need to be familiar with. You need to understand and remember what helicase, single-stranded binding proteins, topoisomerase, primase, DNA polymerase 3, polymerase 1, and ligase, what they all do. Basically, in our diagram, the topoisomerase is going to be ahead of the helicase, and it's going to kind of take the strain off the strand. It's going to kind of unwrap the strands. All right, and as it unwraps the strands, the helicase comes in and actually breaks the hydrogen bonds. Then the single-stranded binding proteins are going to be used to hold that strand apart so that a primer can be laid down, an RNA primer can be laid down by the primase. This is going to allow the DNA polymerase, in this case DNA polymerase 3, to bind to the strand and add complementary nucleotides. DNA polymerase 1 is going to be used to remove RNA nucleotides from the primer and replace them with DNA nucleotides once we're ready to move, and that can be done on the leading strand when we originally lay down our RNA primer, or on the Okazaki fragments at each beginning of the fragment. And then the ligase is, think of it like the glue. It's going to tie everything together to make sure that we have our one continuous strand on each side. Okay, so we can take a look. Notice again the differences between DNA polymerase 3 and DNA polymerase 1. They are both adding nucleotides. It's just about which nucleotide you're adding and when you're adding those nucleotides in. All right, so now we can kind of put that all together. And if this is our DNA strand, the dark blue is going to be our original strand. We can see here that as we add on the leading strand, our continuous segment, it's going to be laid down in a 5' to 3' direction. Okay, 5' to 3' direction here. The little red molecule is representing an RNA primer that had been laid down, okay, by the primase. Notice that as we continue in our right to left direction, we're going to continue this leading strand, but we have to continually add new Okazaki fragments, right, one behind the other, because as the strand opens up, more real estate basically is open for adding the DNA nucleotides. Notice on the other side, when we're going from a left to right direction, the leading and lagging strands are going to flip-flop, because as that replication fork opens up in a left to right direction, okay, this strand can continue to be created, whereas these strands are now created one at a time. Okay, and those strands, again, those little segments are called Okazaki fragments. Topoisomerase is not shown here, but if we had topoisomerase shown, it would be on this side of the helicase, and it would basically be unwinding the strands so that the helicase could then come in and separate the hydrogen bonds between the nucleotides. Okay, as that occurs, single-stranded binding proteins are going to hold the strand open. DNA polymerase is going to continue on the leading strand to add new nucleotides, whereas primase on the lagging strand will add this RNA primer, and then we can continue with DNA polymerase adding complementary nucleotides. When those two Okazaki fragments come together, like you can see here and you can see here, DNA ligase would be involved and responsible for tying that all together. So the polymerase would remove the RNA nucleotide, would replace it with a DNA nucleotide, and then the ligase would come in and basically lock that all together. All right, so the next topic is evolutionary significance of altered DNA nucleotides, and really what that is a fancy way of saying mutations. So a change in the DNA nucleotides, mutations are going to lead to changes in the phenotypes of the organisms. If this mutation occurs in a germ cell, in a reproductive cell, then what's going to happen is we're going to be able to pass that trait on to the offspring. Okay, so this is going to be the original source of variation, and this is where natural selection can operate and ultimately possibly create new species. All right, so now we're going to look at replicating the ends of DNA molecules. And what happens here, the problem is, as you are replicating the lagging strand, you ultimately get to a point where you can't add any more nucleotides because you have to have something for that primase to add to. So what's going to happen here is the cells, or the chromosomes I should say, have something called telomeres on the ends of them, and these are going to be repeating units. In humans, these repeating units are TTA, GGG, and this can be repeated any number of times, from 100 to about 1,000 times. Okay, and so what's going to happen is ultimately, as you replicate the chromosomes, each time you do it, it's going to get a little bit shorter on the lagging end, on the lagging strand of that chromosome. Okay, and so what they think this is for is so that it's kind of a control to help regulate somatic cells and the number of times they can replicate, because once you basically remove all of that telomere, now you start removing genes, and if you're losing genes, the chromosomes are not functioning properly. The cell basically, needs to go ahead and enter into apoptosis, and that cell's going to end up dying off. So we can't continue to replicate that, and that's kind of a safety measure when it comes to cancerous cells. Okay, although there are some cancerous cells that have the ability to add nucleotides to the end, and basically those cells are immortal. An example of immortal cells would be the HeLa cells, the cancerous cells from the individual called Henrietta Lacks. Okay, those cells have been replicating since sometime in the mid-50s, 1950s. So another thing that we need to know about these telomeres is that in germ cells and reproductive cells, there's something called telomerase, which is an enzyme that helps to lengthen the telomeres. So as those cells are replicating, we can make sure that we can extend the leading strand, which is going to then artificially lengthen and allow us to add nucleotides to help the lagging strand to maintain its particular length. And the last thing we're going to look at is something called nucleotide excision repair, and this is basically another job of the DNA polymerase. It is going to proofread and make sure that the nucleotides that are being added are correct. Okay, so what happens is when you get an issue, okay, in this case it's something called a thymine dimer, where these two thymine molecules have bonded together rather than bonding to their adenine counterparts. And so what will happen is the nuclease will come in and it'll remove those malfunctioning nucleotides. Okay, then the DNA polymerase can come back in, add the complementary nucleotides, make the correction, and then DNA ligase comes through and adds the finishing touches, basically ties that all together. So this has been the molecular basis of inheritance. This is your lecture for chapter 16. If you have any questions, certainly talk to me in lecture, and let me know if you have any questions, and I'll see you guys in class.